going to be using some colors. I don't think for this there's going to be any real significance in the colors, other than I try to maintain some semblance of continuity when I'm looking at different things. Yeah. If you were here the other day, you got one. And I do not have any extra sheets.
He wasn't here the other day.
Well, I don't have any extra sheets. In that case, somebody took two and maybe put one in the recycling. I'll have to get you one later on, but that's the way it is. So down below, let's start below and work upward on this page. Everybody is somewhat familiar with the double helix. Okay. Now it turns out that there are only three parts to a strand of there are three components to DNA. One, I'm only going to highlight one of them. One of them is, for example, this guy here. The other are those guys there. And pink, where's my pink? And the other one, pink, is here. Okay. Now what I want to do is I'm going to redraw this up here very, very, very diagrammatically and label them. It turns out that if you look at this, you'll see that this is repeats. If you go down this side here, there's one of these guys, and there's another one of these guys, and another one of these guys, and another one of these guys, and it keeps on going. If you look at the other side, it repeats too, but it's upside down. You see the S here? You see the point, the direction of the point? It's opposite. So in fact, DNA is said to be anti-parallel. It is parallel, but the directionality is opposite to each other. Now you don't care about this until you get to by 12, okay? So just it makes a difference now, but not until by 12. Each one of these is a subunit. Because this is a single repeating unit, we call this a monomer. Mono means one. The suffix mer refers to a unit. So monomer means literally one unit. If you put two of them together, it's called a dimer. Di meaning two, mer meaning unit, so two units. If I string many together, it's called a polymer. It's called a polymer. Poly means many. Mer is, again, unit, so many units. So, for example, carbon fiber nanotubules are polymers, repeating units of carbon atoms. Okay? So each one of these is, in fact, a nucleotide. So the nucleotide, this is the DNA. Sorry, DNA stands for deoxyribo nucleic acid. Okay? A DNA nucleotide, this is the name we give to the monomer. And we're just going to redraw this because we want to be able to identify the different parts. The directionality of the sugar is actually conferred by the presence of an oxygen atom. That's an oxygen atom. And this is a carbon ring, and it is a sugar. And it is attached So, huh? So this guy here, which on down below is a P, this is a phosphate. You guys have run into phosphates when you did chemistry. Phosphate, remember, is the PO4, has an overall charge of -3 on it. So that's what that is. This is a sugar. This, in fact, is the deoxyribose sugar. Deoxyribose sugar. Okay? These guys here are called the nitrogen bases. Okay? And it turns out there are 4 different kinds of nitrogen bases. There is something called cytosine. Notice I'm doing the capital C really big. There's guanine. There's adenine. And there's thymine. Sure. Cytosine, C-Y-T-O-S-I-N-E. Guanine, G-U-A-N-I-N-E. Adenine, A-D-E-N-I-N-E. And thymine, T-H-Y-M-I-N-E. Thymine. Okay? We're going to save a lot of the Y for Biology 12, but if you look below, you're going to notice something very, very interesting. You'll notice, if you go through this list, that adenine always bonds with thymine, and guanine always bonds with cytosine, without fail. Never changes. Dear. That is an oxygen. If you guys remember, think back to what do you call that? Chemistry. When we were looking at organic structures, we said that you could make organic structures like this. Instead of drawing all the C's, you could go like this. You remember that? Okay? Well, this is the same thing. So if you were to actually take this apart, what you'd see is that there's a C here, there's a C here, there's a C here, there's a C here, and up here actually there's a C, and there's one more C, and I'm trying to figure out how that works. Pardon me? It's just oxygen. It's just an oxygen. Oxygen can be embedded in a carbon chain, but that's what it's basically like. And this is a 5-carbon. That's why this is a 5-carbon sugar. Bing, 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 and that's why it's a ribose sugar. Okay. Deoxy just means it's missing an extra oxygen. Doesn't matter. Don't worry about it. But the important part is the nitrogen bases, in fact. Okay? If looking down below, what you'll see is that the backbone of the whole thing, the sugars and the phosphates, they don't change ever. That's why they call it the backbone. It's sort of like this is like the ladder, where the rails are made of alternating sugars and phosphates, sugars and phosphates, and the rungs in the middle, those are the things that change. That is where the genetic code lives. Okay? The order of these nitrogen bases determines the genetic code for whatever gene is present. And that'll make more sense in a second. But it is this order that produces information to be passed from generation to generation. And that's what we're about, is passing information from generation to generation. Now you also notice that there's something kind of interesting down here. And when we talk about each one is a nitrogen base, because cytosine always bonds guanine, we consider these a base pair. Okay, we call these a base pair. Okay, adenine, thymine are another base pair. All right, and the last notation I want to put on here is that we do have a directionality in this. We have, as it were, DNA having a directionality in this direction, and DNA having a directionality in this direction. And the word that I used before to describe this is anti-parallel. Okay? So, does any of this ring any bells for you? For some of you, it's, yeah, I remember this, I'm starting, okay, kind of, okay. Okay, yes, and Ann. I'm sorry, say again. Oh no, that's just. I had a very sad childhood, so I color a lot now. Apparently, I found this out today, that sarcasm is a Canadian thing. This is true.
Serious?
Yeah, sarcasm is actually a Canadian thing. So if you go to the States and use sarcasm, they're going to look at you funny. Like,
Oh wow, I totally didn't know that.
At least you guys get proud of something.
yeah.
Oh,
Sarcasm, invented only in Canada.
Get to have it, and it's widely popular.
It is. But apparently, you'll get this. You guys have not talked to Darlene yet. She was the one that told us this is actually true. That if you use sarcasm in the States, they won't get it. If you use sarcasm in most other languages, they won't get it. Which is really weird, because my wife and I, when we were going out, we would be so sarcastic to each other. My mom used to rag on me like all the time. She says, "You can't treat Rhea like that." And he was like, "Mom, we're kidding. Relax, we're kidding." We're not sarcastic anymore. Apparently there's a cutoff point. I'm just saying. Okay, so okay. Here's another diagram that you should probably have seen at some point before. And the reason why I put this in is so that when we talk about different terms, you'll have an idea in your mind of, oh, I think I know what that he's talking about. First of all, chromosomes. This is a chromosome here. The A is a chromosome, but you only see chromosomes like this at a certain point in a cell's life, and this is at a time called prophase. So this guy here is a prophase chromosome. For most, thank you. This is a prophase chromosome. For most of a chromosome's life in a cell cycle, it just looks like one giant strand of spaghetti. Okay? And when it's like that, for 99% of the cell's life, it's called, depending on who is, you're American or Canadian or whatever, some people call it, I call it chromatin. I've heard people call, Americans call it chromatin. So you choose, I don't care. So this is what we call a chromosome. Okay? This here is just an area. So it's a specific area of a chromosome, and this is called a gene. Yeah? The line. Oh, this, okay. Okay, pretend this is the 99% of a cell's life. Okay? This is chromatin. And it's made up of all these little beads. And each one of these beads, for example, is a pair of nucleotides run across them. So this is one big, ginormous strand of DNA. Okay? And it lives like this. It has to live like this for most of the time. But when it wants to replicate, what will happen is this will quickly double, and then it will start to condense. It'll start to wrap around, and you've got to pretend that I know what I'm doing here. It's the first time I ever tried to wrap this thing around, and it's not going well. I can feel it. It's just making a ball in my hand. But if you can imagine it shortening and coiling and supercoiling and coiling upon those coils, what you'll end up with is this guy here. That's why this only exists at prophase. Okay? The next thing that will happen is this will split. This will split in half right here. One cell will get this. The other cell will get that. And that's how you can duplicate cells. Okay? So I don't know if I want to talk too much about that. We're more interested in the information that is transferred from this guy and the information transferred from this guy in the other direction. That's what we really want to know. Okay? So a chromosome down below, I'm going to be going back and forth. I'm going to refer back and forth to the diagram and the description below. So a chromosome is a macromolecule or polymer. A polymer macromolecule. Macro means like ginormous. But because it consists of consecutive chemical bonds, it is still considered a molecule. A really ginormous molecule, but it is. Okay? Of thousands of nucleotides. Containing, thank you, whoops, that was too far. Still too far. Okay, containing many bits of information. Okay? So this A here refers to the A in the diagram up top, okay? So B, if you look at B, I'll just spruce up a bit. B, this is just a section of the DNA. Okay, so it's one section of the entire chromosome, and this is called a gene. Okay? So a gene is an area of the chromosome containing information for the manufacture of one protein, for the manufacture And biochemistry often talks about this in terms of the one gene, one protein hypothesis. It's one of the central tenets of all biology. Okay? Okay, now we look down, we see we've got we understand what the chromosome, it's the big giant, the whole thing, the whole enchilada. Yeah, so Mexico. The gene is just one piece of it, and it codes for a certain gene. This whole C is just meant to represent this is part of the DNA strand. And so we say this is a double, comma, antiparallel helix. Helix means ribbon, by the way. Yeah. Hypothesis. Yep. Where are you? Do you want the spelling of hypothesis, dear?
Like right after the O, I couldn't read that letter.
Oh, T. It's HYPO THESIS. Not at all. Please don't feel funny asking me because I know my writing's so bad. Helix, composed of nucleotides. Okay? Oh, gee, sorry. And you know what a nucleotide is now, okay? So there are a number of mistakes here. So we'll deal with them. So that's C. D is not a nucleic acid. DNA is nucleic acid. D should be a nitrogen base. Okay? So it's just pointing to a nitrogen base. And this is simply a ring. Can't spell either. A single or double ring. Why do I keep putting a W? Ring structure. It has been a long day. Containing nitrogen. Okay? And these, the order of these determines the information contained. Yep, thank you, dear. Okay. The sugar phosphate, the ribosugar phosphate background, this only provides structure and protection for the nitrogen bases. It's like you have to hang the nitrogen bases on something, otherwise it's going to be a soup. You can't have a soup. It has to be in an exact, precise order that does not change. So you need something to hang it on.
And like structure and what?
And protection for the nitrogen bases. Okay? Okay, we good with that? Okay. Why DNA? Turns out that points to ponder we don't know exactly how many, but we do have what do they call it? Not a baseball number. Ballpark. That's it. That's it. Ballpark number. We do have a ballpark number of how many different kinds of proteins there are in the human body, and it turns out to be about 100,000 different kinds. Okay? Ponder. Ponder meaning think. Hey? Okay. About 100,000 different proteins. In the human body. And they fall into about three or four different really roles. The first role is that proteins are used to build structures. They're parts of, they constitute every organelle in a cell, and they're part of the cell wall, in fact, to build structures. And other things, like hair is actually it's called keratin, it has keratin in it, which is a protein. Nails are too, all sorts of things. It also has, the other thing that they're used for is things called enzymes. These control all cell function. And therefore, all life. There's a third thing that proteins are used for, and that is a certain classification of hormones. And hormones are considered the most powerful molecule on the planet. No, seriously, no, you're laughing. I was trying to make a joke. It's actually factual. If you look up deadliest things, it's actually a protein that is. But I'll tell you this. No, I'll save that story for file 11. Okay, hormones. So these are chemical messengers. chemical messengers that are made in one part of the body and are used in a different part. My Bi 12 students are really finding that out this year. I hate to be a biologist at that day. Okay? But it does have some other things as well. It is involved in immune system. These are antibodies, things that fight disease. What else? I don't think that's necessary. Okay? So these are the four main things that they're responsible for. But overall, all of these things together, these constitute antibodies. A-N-T-I, antibodies. All of these things together produce an individual's characteristics. Okay? Not only the characteristics that you can see, like hair color, eye color, skin tone, and that, but how your body actually works too. Okay? All of these things fall into this category. Eman, did you have a question? Okay. So, I was going to say something else. Can't remember. It'll come back. Okay, this what you see, okay, down here, proteins in the body. Give some examples. Do I want to give some examples, or is this too much? No, I'm going to leave it, because you don't need to know any of that. So we're just going to scratch that out. I don't need to know that, because I've got that. Okay, this is where it starts to get interesting. We're going to talk about Gregor Mendel. Gregor Mendel was a monk who was Austrian, I believe. Gregor Mendel was Austrian, and so he lived in a monastery out in the mountains, and he didn't have a lot to do, and he was the gardener. Yes. Gregor, just as it is. Gregor up here. Gregor Mendel. Anyway, he was the gardener, and he noticed some things about pea plants that were kind of odd. That they came in different, not only did the flowers come in different colors, but he noticed that sometimes the pea pods were skinny, and sometimes they're fat, and sometimes the leaves were in different places. Axial meaning near the stem, others terminal on the ends. And so he started to wonder about that. Why is that? Before Gregor Mendel was around, it was thought that it was recognized that there are characteristics that you see in a child that you would see in the parents, but it was kind of like a mix, and they didn't understand that. They thought it was kind of like an equal dilution, and they had a lot of different understandings. But they understood that in order to produce a child, the couple would have to have sex. Okay, we get that. And they also understood, at least the idea, that the males produce sperm, and after the sperm was implanted, that's when a child would be conceived. They got that. But what they couldn't get was, how is it that this human resulted from this? Didn't make sense. So they actually had, and you can look this up online too, they also thought that the baby was actually within the sperm. It's called a homunculus. That's what they thought. And you can look online and you'll see drawings of how they conceive things to be. Well, what Gregor Mendel Gregor Mendel and Charles Darwin were contemporaries, but they didn't know each other from a hole in the ground, because Gregor Mendel was living in the Alps of Austria, and what's his name? Darwin was living in England, and so they didn't have much to do with each other. Okay, so anyway, he started to list the different types of characteristics that he saw that were different between the two. And he came up with, I think there's 9, I think he figured that, something like that, 9 different characteristics. And then he started to experiment with them. And that's where things started to really take off for the understanding of transmission of information between generations. So, let's get a little bit of notes in, and we're going to get right into genetics. Thank you, Jesus. We have about half hour. Okay. Prior to Gregor Mendel's experiments with pea plants in the mid-19th century, it was thought that offspring would show a mixture of both parents. And you guys, some of you, if you think about your own family right now, some of you will recognize that a child may be more like his father and less like his mother by quite a bit. Which was, they couldn't understand that because they thought it was an equal sharing of traits, and it's not, of both parents.
You spelled it with a B instead of a P.
All right, leave me alone. Okay, so for example, let's not even look at humans. They thought was this, that if you had a white flower and you crossed it with a red flower, you'd always get a pink flower. That would be their expectation from this, that it was an equal mixing or merging or dilution of the two parental traits. Okay? But what happened was just the opposite. Now, that can happen. Don't get me wrong. Carnations do that. But he found that if you only one of two traits would be present. So what he found, this is what it was thought. For example, this was the thought. This was the thought at the time. What he discovered, though, is that you could have Mendel's experiments showed that you could have a white flower and you cross it with a red flower, and you'll get a red flower, which was not what he expected, because he thought that too. up till he started to do the experiment. Another example was seed shape. Okay, he found there's a wrinkled seed and there's a round seed, and the thought was like if you mix these two, you should have got something in between. But what he discovered was that the round seed always overshadowed it. Always, if you cross those two, the round seed would always be the offspring, always. Okay? So when he did that, if that occurred, if there was one trait that always seemed to dominate, he called that the dominant trait. And he called the trait that was hidden or overshadowed, he called that recessive. Recessive means to recede, means to shrink into the background, so to speak. And that was actually a very good word for him to use, because it was hidden, it was shrouded, it was cloaked. So in the, we're only looking at 1, 2, 3, 4, 5, 6, 7 traits. So the seed shape, we found that dominant was always round. We already said that. Recessive trait was always wrinkled. The seed color, dominant, would be yellow. And the recessive would be green. Notice that I'm using capital letters here and lowercase letters here. Again, this is just my way of kind of showing who is on top of this thing. So pod shape. So you know, you're talking about the shape of the pea pod where all the peas line up inside. The dominant trait was inflated. Those are my favorite. At least you get something out of them. Whereas the others, the recessive, was constricted, skinny and shrunk. The pod color, though, the dominant pod color was green. And the recessive pod color was, what do you think? Yeah, it was yellow. The flower color, dominant flower color, was purple. Recessive, white. Flower position, I'm going to use simple words. Side. So at the side of the stem, like if this is the main stem and you've got more things going off.
What's the action?
Axial. Okay. The actual word is axial. If you look at the recessive trait, the flowers would be on the end, and those are called terminal. So these are on the end of the stems. Okay. The last one, stem length. So how big is the stem? The dominant was always tall. Recessive was always short. Okay? To be honest with you, I can't recall. I don't even know if I ever really knew, but how long Mendel was doing this for. The upside of peas is that in a given growing season, you can cultivate them, recultivate them, and do it multiple times. The other thing too is that between growing seasons, you can store the seeds without losing them. That they'll be good the next year. Okay, so I'm sure he did this for a number of years before he even considered publishing. But when he published, this is what he said, and these are Mendel's laws of heredity. Now. You should have some kind of handle of what Mendel stated. And there's only 3, so it's not really hard. And if you think about them, this is logic. This is not rocket science. It's logic that we now take for granted, but back then was earth-shaking. The first thing was that inherited characteristics are controlled by what he called factors. We now refer to those as genes. He didn't know what a gene was. He had no concept of a gene, but he just knew that there was something there, and that each parent donated one factor or gene. Second statement is that one factor dominates the other factor. I'm just going to put the other. And one factor is masked. So now we know those as dominant and recessive, which we've already mentioned. It's dominant and recessive. Okay. Now this next statement is the earth-shaking one. Okay? This is pretty straightforward because he noticed that. But what he also noticed was that, for example, if you had a trait that showed up in the parent generation and it disappeared in the next generation, it could reappear in the second generation after that. So that's why he said that one was masked, meaning it wasn't destroyed. It was still there, but you couldn't see it, and it could show up later on. That was the earth-shattering thing, right? Nobody ever noticed that before. Okay? Now this is where it gets. This is incredible, and I'm going to use the word genes. He did not use those words, but I'm just going to modernize it for this. Genes separate during sperm and egg production. And only one of the possible traits is passed from each parent. Okay. Okay, we'll look at this again and we'll define it again. But in a nutshell, like looking at this, it's like, wow, that's a mouthful, man. Like, what does it mean? Basically, what he was saying is this: is if you have this is one parent, or no. No, let's not go there. Some definitions. If you're given, first of all, in order to do this, we need to establish something. So if we use tallness and shortness, the factor or the gene for tallness, we're going to make, this is a capital T. So this is going to be tall. And for shortness, we're going to make it small t. That means short, okay? And so in his mind, this guy, because of his, remember he looked at those different traits? What he was saying was the tall dominates the short, okay? He's the dominant, short is hidden, okay? So when we're talking about genotype, what we mean is what is the actual gene combination. From both parents. So for example, if this was the father was tall, so I don't know if you know this, but this is a symbol for male, symbol for females like this, okay? If an individual received one tall gene from the father and one short gene from the mother, the actual gene combination is going to look like this. So this, capital T, small t, or capital T, capital, whatever combination of letters, that is considered the genotype, okay? Phenotype is what we see on the outside. So in this case here, if we're going to be looking at this individual, what would we see? What kind of individual would he be? He'd be more like the father because it's dominating. So, this dominates this. So shortness is hidden, tallness is dominant.
But does male always?
No, it's not male. It's not the male. It's just that's just the way it worked out. I could have turned this around and made the female have the dominant allele for tallness and the male recessive allele. I had a friend in high school. His name was Len Soot. He's a Dutch guy. Both of his parents, I met his parents. His parents were like this tall and skinny like this. Len Soot in grade 8 was as tall as I am, actually taller than me. He was 6 foot tall, and he looked like Arnold Schwarzenegger in grade 8. In grade 8, he was like this.
What was his name?
Smith. Len Soot could leg press 600 pounds twice. And in those days, they didn't have leg press machines like you have at the gym now. If you wanted to leg press 600 pounds, you put a bench under a rack, and you put up 300 pounds of plate on each end of the bar, scooch your butt down to under thing, and push straight up. He did it twice in grade 8. When I met his parents and his sister, Marion was just this sweetness of a girl that if you blew at her, she'd probably fall over. And the parents were little wee, teeny tiny things. Leonard, where did you come from? But it's because of this dominant recessive situation. No, he wasn't. He wasn't adopted. I'm still Facebook friends with Leonard. There's a little guy, I gotta tell you this story. This is the best story ever from my high school days. By the time we got into about grade 10, oh, your grade, there was a little British guy who was like Len's sister, was a little wee wisp of a guy named Davy Smith. Had a wonderful British accent, but he's frail. You looked at him and he was very frail. And like you know what Leonard was like. He was this there was an unwritten rule. don't ever touch Davy Smith, because Leonard will crush you. You will not recover from the beating that he's going to lay on you. Literally, Leonard played football. The school I went to was ranked in the top 3 in the province every year. When you put a helmet on Leonard, he changed from a gentle giant into an animal. You could see the change in his eyes. His eye, all of a sudden, he was amazing. But he was the nicest guy you would ever want to meet. And he was the nicest guy you'd want on your side. And it was really funny because in my grade, in the whole school, I can only think of these 3 guys that were troublemakers at all. And one day they made the mistake of getting too close to Davy Smith. Right away, you could see this bear. Grade 8 comes up and you can see this. It was so funny. The whole school knew what was happening. The teacher noticed it. And he said something, I never forget, he said, have you ever noticed that the guys that really are tough don't have to say they are? Because Radikoff and his other 2 guys, they were always trying to push themselves around, and they are just nothing. Leonard, this guy was tough. He never had to raise his voice. He was mild. I never heard a bad word come out of Leonard's mouth. What a guy. Leonard was amazing. And he was the protector of those who were weaker and could fall victim to abuse. He was their protector. And nobody, I tell you, nobody would dare even look sideways at Davy Smith after that. It's like, okay, I know my place. Anyway, what we see on the outside. So what we'd see, if an individual had these 2 copies, we'd see that this is tall, because this overpowers that. Homozygous is when both copies are the same. In other words, they're either one from father, one from mother, they're both tall like this, or they're both short like that. No, they're not twins. Every person, they're like twins in a way. They're like genetic twins. But every person in this room, you've got 23 pairs of chromosomes. So for every gene, there's at least 2 possible outcomes. Do you know? Here's a thought for you. When your mother and your father conceived you, do you know how many possible combinations could have resulted? No, not that many. 15 million. There could be 15 million different combinations of their DNA to produce you. Crazy. If something is heterozygous, that means when you have one of both, or have one of each. One of each. So heterozygous, something that's heterozygous will look like this. Capital T, small t, for example. Now, I've held off using this word here. And again, it depends on who you talk to, how they pronounce it. I called them alleles, but I've heard it say, I think Hank on Crash Course called, what's that Y sound? No. It's alleys, I yield. I don't know. He says it funny. I don't know. Anyway, this is a possible variation. So, what do we mean by that? Each one of these represents, in actual fact, you have one allele from each parent. So that is a possible variation. That the capital T is an allele, and so is the small t. So that is the possible combinations. Okay? Are we okay with that? Now, how does this work out? Now, this diagram, let's suppose that these are two chromosomes. Okay? They're two of the same type of chromosome, one from one parent and one from another parent. Now, this is a make-believe organism. It's not even human. Okay? So, if you can tell that these are the same chromosomes, one from each parent, because they're the same length. This thing here, this knot called a centromere, is in exactly the same place, and you see the banding pattern is identical. Okay? Now, probably we would have been better to there's different ways of noting allele differences, and one is having them plus, and other ones are capital or lowercase. So if you read the caption here, for example, these two homologous chromosomes, homologous means same, have 4 genes for the same traits. A stands for abnormal toenails. B for blue blood, C for crinkles, and D for dog breath. Okay? You're wondering, what kind of critter is this? So am I. Okay. If you have a positive sign, this means that it has this condition. If you have no superscript, it means the condition is absent. Okay? So what does this mean? This means that A is for abnormal toenails. So this guy has got abnormal toenails. Okay? This guy doesn't. B is for blue blood. So this guy's got blue blood. I suppose, as opposed to red blood, mind you.
Wasn't that like an old myth about like royalty? Like, I think there was some old myth that like the royalty was born with blue blood somewhere.
I don't know how the thought of blue blood comes into it, because there is, I don't know. Let's Google that sometime, okay? C means for crinkles. And D plus stands for dog breath. So in this case here, you have two individuals. So crinkles, or let's say smooth, normal, and I'm going to say D for dog breath. Nobody's got dog breath today. Okay? Thank God, eh? Jeez. Give them a certs. So what you see here is two chromosomes, one from each individual. One individual would have abnormal toenails with crinkles and red blood. The other has normal toenails, but he's got is smooth, but has blue blood. These are called alleles because they're the possible outcomes. They live in the same place on the same chromosome as the other alleles. Okay? Dominant alleles from now on are going to be capitals. Lowercase is always going to be recessive, and that's how we're going to do it. We're not going to use plus or minus, we're going to use dominant, okay? So in the case of, if we're going to talk about tallness, just like we said before, T, pure breed T is going to be the tall allele. Lowercase t is the short allele. Pure breeding, this is another term that should probably have been on the other page. Pure breeding means either pure breeding tall will be this, pure breeding short will be this. So you'll notice that pure breeding means homozygous. Another way of saying it. Okay, now this is where we start, the rubber starts meeting the road, or the road. Okay, let's say a pure breeding tall plant is crossed with a short plant. What does that mean? That means that the parents are this. Please note how I do this. Parents, one parent is going to be pure breeding tall, and the other parent is short. For that to occur, it has to be that and nothing else. Because if there was a capital T, it'd be tall. The question is, if you cross these two, what are the individuals going to look like? So this is how you do it. Because of this situation where every person has got two chromosomes and you have alleles for different traits on both, you have to write down the possible alleles. But we don't call them alleles, we call them the gametes. What is going to be in the sperm cell that is lucky enough to hit the egg, and what is in the egg that's lucky enough to be hit by the sperm cell. So what we put next are the possible gametes. And that's easy, because in this case, the possible gametes are this, comma, this, and this, comma, this. So what we do in order to calculate this is we invoke something called the Punnett square. And the Punnett square is almost like a tic-tac-toe, like this. And we're going to put one parent on the top. We're going to put one parent here. The gametes. Don't mix the gametes. So if I'm going to put this parent on the top, the gametes are going to be like this. One gamete per square only. Okay? Down the side, one gamete per square. Now, where these boxes are intersected, that is where you put the alleles from each parent, or the possible alleles from each parent. So in this case, oh, and by the way, you always put the capital letter first. Okay? So this is going to be capital T, small t, capital T, small t, capital T, small t, capital T, small t. So I will ask you, and this is the type of problem you're going to get, give me the genotype. I'll finish with this. Thank you. Of the genotype and the phenotype of this cross. The genotype is the actual genetic makeup. In this case, the actual genetic makeup was 100% capital T, lowercase t. The phenotype was 100% tall. Why? Why is it tall? Dear. Yeah? Thank you. That's exactly right. Why? Because the capital T dominates the recessive allele, and that's why it's tall. Okay, we'll leave it there. Now, you should, I think you can do 121 or 1.2.1. That means that I'm sorry, 2.1.1. So 2.1.1 is here. Just do 2.1.1. I want to spend more time playing with this before I turn you loose on 2.1.2, okay? So we are a little bit behind, but we will try to catch up tomorrow. Love you, go away. Oh, just go ahead and do it.
Yeah, I've already seen all the answers for it. Because we-
Okay, then can I just did I give it back to you?
Yeah, I have that.
Can you just give it back to me? What I'll do is I'll recalculate. So what I'll do is, like, instead of making your test out of whatever it is out of, I'll make it out of what I'll just basically eliminate those and then recalculate those. Okay. There's no such thing as that. Oh, wait a minute, that is no.
Yeah, no.
Thank you.
Okay, how did you know that these are listed?
Doesn't matter. You can put it anywhere.
But like, how did you know that these are two capitals and these are two?
It's given. This is just like a sample problem.
Okay, but then how would you know if you were getting it out of a question?
Oh, you will.
Which would,
Like in
okay,
the
should I
pre-
just ask you then?
Oh no. We'll do some tomorrow together.
Okay, because we did this in Bio 11 and I didn't understand it. I really got more upset.
But you know what happens is that often it's like hammering nails. Nobody ever drives a nail home on the first throw.
That's not true.
Unless you got an air nailer, then bang, you're in. But usually it takes at least 2 hits before you get it. Okay, so don't feel bad about that. Yeah, that's why I want to tomorrow start you guys off at 2.1.2. Do a couple of them, and then okay, I get it.
So what is it
I'm going to start you on this. What probably I'll do is, what I'll probably do is we'll do a couple, and then at that point, hopefully everybody's kind of like, okay, I get what's supposed to go on here, and then I'll probably let you guys. I want you guys to do some of them on your own because I want you to get handy with them,
Okay.
because that's a lot of what we're doing.
They've been working with cross species.
Oh, might be take a while.
Guess what they made?
What?
Pink blue.
I could do that with shoe polish. What?
In chemistry, I was doing the things. Is hydrogen, when you do the IDE, it's not hydroxide, it's hydride?
Hydride, yeah.
Okay.
Like it was, say C.
Have a good day.
CH3, that'd be say, or CH4. We call it methane, but really we call that carbon tetrahydride. That's correct. Yeah. It's always, if it's binary, it always ends in IDE. Yeah.
Okay.
Yeah.
So it is here.
Yeah.
Is that not enough to get the functions?
Let me know. I just learned. A compound is.
Because I didn't get the chance to have this. The first thing the market just put it.
The problem that I have is that it's varying ratios, and the point,
They're in fixed.
they're in fixed ratios, that's where I probably, like, so far as the first part of that, I'd say that that's good. How about we do it this way, take a half more for that?
Yeah. I did it down here. I did it there, so with that.
That's fine, no, that's fine. Oh, yeah.
Okay.
Yeah, there's more than one way of skinning that cat.
How did I lose my ears here again?